Hey there, this video is going to cover a pretty common recipe, which is multi-condition comparisons, especially when looking at bulk RNA-seq or other kinds of omics datasets. So we'll be using this bulk RNA-seq dataset as an example that we'll be walking through. And what you can see when we look at the sample annotations for this data is that we have a couple of different treatments. Specifically, this 0% ethanol condition will be our control or vehicle here. And then we have two different levels of ethanol treatment, this 0.08% and 0.16% ethanol. So different levels of treatment that we might want to be comparing again against this vehicle control. I also have some metadata around the donor that provided the primary human hepatocytes that went into these different samples, and then a little bit of metadata from the published dataset in this case. So to begin our analysis, I'll head over to the Analysis tab. We'll show some analysis on the canvas as well later in the video. But just to keep things simple for now, on the analysis page here, you can see that we have a blank slate where we can really get started performing our analysis. And here we'll just start by opening the analysis modal and selecting differential expression. I'll go ahead and create an analysis here, and then set it up for the first group or first condition that I'll be looking at. So again, referencing the treatment column here, I want to group my samples by treatment. And my first experimental group will be this lower ethanol condition compared to the 0% ethanol. And I can click Run for that analysis. And that will kick off our differential expression analysis bioinformatics pipeline, which will load by default as a volcano plot. I can customize that plot, such as changing the statistical significance and editing different plot appearance parameters like the color of the plots. I like to follow a particular palette for my volcano plots here. I'll do that and then select a different significant up and down values here. And then you can see I can customize the labeled gene colors also. I'll just make those different for the up and down. So I'm just changing these colors right now in the legend, and you can see a preview of that. And then when I click Save Changes, all of those changes will be applied to my volcano plot. So here I can highlight some genes of interest, these ones that are showing particular significance values. Great, so that's a nice looking volcano plot for my first condition. I can exit out of that full screen editing mode and see that I have this particular comparison with my nice volcano plot. So we know that we have another comparison we'd like to do in this data set, which is that 0.16% ethanol. One nice shortcut here is to just clone the volcano plot that I've already created for the first comparison. This gives us a great starting place for doing the second one. Now I have two copies of the 0.08% ethanol, but I can edit this one, go back into the analysis, and select the 0.16% ethanol group instead, and click Run. This now updates that volcano plot, showing the results instead for the 0.16% ethanol. So here, interestingly, you can see that because I highlighted some of these genes on this first condition, you can see the similar genes that are highlighted in the second group. We can reorder these plots on the page, so if I want this lower ethanol version on the left and higher on the right, I can do that. But with our labeled genes here, we can already see that we have some similarities across the two different conditions. With a multi-group comparison in Pluto, what we're really asking, or going to be asking, is how many of the genes in totality are overlapping between these two conditions. So a common way to address that question would be to use a simple Venn diagram to look at all of the genes in this condition and all of the genes in this comparison. I can do that again from this analysis page. I'm going to move over to the canvas because I like the visual aspect of seeing the overlap. So on my canvas here, I'm going to bring on my two volcano plots. And kind of have them shown here stacked on top of each other. Great, so I have that lower ethanol condition here, the higher ethanol condition here. And really what we want to be doing now is drawing a Venn diagram that's going to be overlapping the differentially expressed genes in these two different comparisons. So to achieve that on my canvas, I'll create a new analysis and select the Overlap Genes option, create analysis, And now I can add different lists or comparisons here. So for my first list, I'll be using the differential targets from this lower ethanol condition. And so I'll just call this one 0.08% ethanol, name my list that way. And then I can set different thresholds for the significant genes that I want to use. So based on that volcano plot, there were quite a few significant genes. I might want to use a pretty stringent p-value here. And I could be comparing just the upregulated genes, just the downregulated genes, or both. I like keeping that comparison directional, so I'll just say the upregulated genes here. We would want to use similar thresholds, likely for looking at this higher ethanol comparison. So I'll name this list my 0.16% ethanol. Use those same p-value thresholds and look at just the upregulated genes and click Save. So now I've built these two lists that I have here. I could add another list if I had another comparison, or if I had a known gene list or gene set that I was interested in using as part of my overlap. But I'll just go ahead and click Save. Okay, so now we can see based on the thresholds that we had chosen that 166 genes are in common between these two comparisons. I can edit the Venn diagram here to show the list name instead of just the number. This is nice so that we can visualize that. And then I can change the color of these different overlapping. So maybe choose some different colors there for my two circles. Exiting back out onto the canvas here. Again, this is why I like this visual, is because I can draw some lines here indicating the comparison that we just did. So now what we did was take these two different differential comparisons with higher and lower ethanol concentration and then perform an overlap based on certain p-value thresholds and fold change thresholds to identify 166 genes that were in common between those two. Going back into my results here, I can look at the results table to actually pull out the gene symbols in those different overlapping sets. So here you'll see that in Pluto, these gene lists are truncated, but you can copy these out to use them into bar plots by downloading that full CSV. Thanks for watching.